March. And then we were going to do it in person, but then COVID hit us and we had to cancel. And then the next time we scheduled it for the end of August, and we had the wildfires and we had to cancel again. So I'm really excited that today this is working and nothing has happened, and I hope it stays this way for a long time. As Nick said, I am Raquel. I am an assistant professor at UC Santa Cruz in the Department of Microbiology and Environmental Toxicology. And I'm originally from Spain. I did my PhD there, and 10 years ago I came to California to do a postdoc in UC Irvine. And in 2019 I started my lab, and I'm going to share with you the research, part of the research that I did as a postdoc and the research that we are doing right now in the lab. I see a few names here that I know, so that is exciting. People from my lab are here, so I'm excited to see them here. But I also see other people from the university that I know, and I'm excited to have all of you here. So I'm gonna share my screen. And I hope you all can see it. I was telling Nick that I like to use this picture, but I guess, most of you, if not all of you, are locals, so this picture is not so impactful. But I really like, it's really amazing just to realize that I live here and that I can see this sunset like pretty much every day. And the work that I do in the lab is related with how environmental pollutants affect metabolic disruption in human populations. So that is why I decided to use this title, To Eat or Not to Eat. Is that the only question? Because when we think about metabolic disruption, we generally think about eating too much and not exercising. However, now there is evidence showing that there may be other elements in this puzzle that are playing an important role in the current metabolic diseases that are all around the world. And in particular, I am focusing on transgenerational epigenetic inheritance. Okay, so just to start, I would like to introduce you to the exposome, the concept of the exposome. So it is defined as the measure of all the exposures of an individual in a lifetime and how those exposures relate to health. And this definition comes from the CDC. And it refers to all the, I'm sorry, I'm having issues with my, okay. So this exposome includes all these different factors that we are exposed to, and that includes pollutants, like here represented in the right side of the picture. But there are other elements like our social environment that is actually related with environmental justice, or our lifestyle that can be related with life choices. However, that is a little bit controversial since there are certain moments in life where we can actually choose, we cannot choose we cannot make these kind of decisions. And also the ecosystem, the region where we live at, if it is a very urban area, or if we live close to a wild environment, that can also affect our health and in general our well-being. And if all these different factors are not enough, these factors also interact with our genome. So we have very unique genomes. Each of us is slightly different. We are all humans, but we are not exactly the same. So depending on how our genome is written and how these factors are interacting with this genome, we are going to develop certain diseases or to have a higher predisposition to certain diseases. So I would like to introduce a few concepts in toxicology. So toxicology is the branch of science concerned with the nature, effects, and detection of poisons. And poisons are substances that cause death, injury, or harm to organs. So these effects, like death, injury, or harm to organs, are like very strong effects. So it seems that based on this definition, we need to look to very dramatic effects to actually consider one, factor to be a poison or to have a toxic effect. So there are some examples in our life that depending on the dose that we are exposed to them can be detrimental to our health. So we are all familiar with alcohol. So if we drink a lot of alcohol in a short period of time, we can die. Caffeine can also be a factor that can kill us. In this case, we need to drink a lot more caffeine than what I think is possible, but it can also lead to death. And even water, that is something that we all need and that we need to drink certain amount every day to actually support some of our biological functions. If we exceed certain levels, that can also lead to death. So the way it is defined, the capacity of any factor, any component, or any chemical to how these factors are considered lethal or toxic is by using this measure that is called the lethal dose 50% or LD50, that refers to the dose at which 50% of the individuals die. So keeping this in mind, this actually supports the idea, the toxicology dogma of the dose making the poison. So every component in our life, including water, that is something that we need to live, can become a poison if we are exposed to these factors in a certain level. So what does this mean? So if we look at how the individual's response to certain concentrations of whatever chemical you are interested in or whatever factor you are interested in, we generally represent it in this type of graph. So in the x-axis, in the horizontal axis, we can see the dose of this factor in an increasing manner. And then in the vertical axis, you can see the response in terms of death. So the LD50, the lethal dose 50%, will be this point where 50% of the population will die, and it will be at this particular concentration. However, there are other measures that are also important. And in this case, they are indicated here as the low IL and the no IL. The low IL refers to the lowest observed adverse effect level, or the lowest dose at which adverse outcomes have been reported. So still at this concentration that is a lot lower than the LD50 is showing adverse effects. And adverse effects in this context refers to death or reproductive toxicity. The no IL is the no observed adverse effect level, or the highest dose at which adverse outcomes have not been reported. So again, adverse outcomes, death, or reproductive toxicity. So, however, something important is that even though if you haven't seen any individual dying because of the exposure at this concentration of whatever chemical you are interested in, that doesn't necessarily mean that this individual is completely healthy. So a lot of policymakers in industry, they use the NOAL to determine safe concentrations for human exposure. So for instance, if you are thinking about a chemical that can be put in plastics to make the plastic more resistant to heat, so we can put it in the microwave, they are going to be looking at these NOAL concentrations. So they are going to say, okay, there is no report that those levels are going to be negative for human health. However, if they are only looking for death or reproductive toxicity, that doesn't necessarily mean that those doses are safe for other endpoints. So this is important in our field because this connects with endocrine disrupting chemicals that are a group of chemicals that I work with. And these chemicals are defined by the Endocrine Society as exogenous chemicals that interfere with any aspects of hormone action. So what does this mean? So these endocrine disrupting chemicals, or EDCs, affect the endocrine system. In the endocrine system, we can find different hormones that are regulating very important biological functions like reproduction or metabolism or behavior. So if we alter the activity of these hormones in any way, we can have a detrimental effect. They can lead to infertility, they can lead to obesity, diabetes, or behavior issues. So hormones act in very, very low concentrations, in very, very low amounts in our body. So we don't need really high amounts of EDCs to actually alter the biological function of these molecules. They can work at very, very low concentrations. And in many occasions, these concentrations are lower than what the industry is defining as concentrations that are safe for humans. So when did this all endocrine disrupting chemical field started? So back in the 60s, Rachel Carson published this book, Silent Spring, and she described for the first time the effect of these environmental factors, these EDCs, in wildlife. And a few years later, in the 90s, Theo Colborn and collaborators published this book, Our Stolen Future, that became a bestseller in the United States. And they were actually describing the effect of these factors, these chemicals, in human health. And after that, there have been a number of books related with the effect of these chemicals in our health. And each of them is exploring these effects in from different angles. So what kind of effects EDCs can cause? I already mentioned this a little bit, but EDCs, exposure to EDCs during pregnancy can lead to adverse birth outcomes. So they can lead to preterm birth or to low birth weight, and that can actually lead to further adverse health outcomes later in life. They can lead to metabolic disruption, either during adulthood or also exposure during pregnancy. They can also lead to behavioral problems, autism, or altered child cognition, again, depending on the window of exposure. And also they can affect reproductive health. These are like the main fields that are being studied in the field of endocrine disrupting chemicals. However, there are other adverse health effects that are also associated with exposure to these chemicals. So my lab and the work that I did during my postdoc and the work that we are working on right now is related with metabolism and the association of EDCs with obesity. And why is this important? So if we look at the map, the world map of obese population back in the 70s, you can see that all over the world, the average obese population was somewhere between 10 and 15% of the population. So, you know, it is an important percentage, but if you look at the same map a few years later, like in 2016, you can see that these percentages are going up and it doesn't seem to be going down anytime soon. So for instance, in countries like United States, they have more than 50% of adults considered obese, and actually there is a high percentage of infants and toddlers that are also obese. And that is concerning because there is a strong association between obesity during childhood and obesity during adult life. And obesity is a global burden at many levels. So on the one hand, we have a health concern. So obesity is associated with cardiovascular disease, diabetes, different types of cancer. It can also affect economy. So there is an estimation of $2 trillion spent worldwide to treat obesity in terms of direct costs or indirect costs every year. And it also has a social burden in that developing countries cannot afford the costs. There are racial disparities, so there are not all communities respond the same way to similar environmental factors. And there is also a lot of stigma. Obesity is generally not well seen in the current culture. So it is important to understand what are the factors that are contributing to obesity? So we have genetic factors that are known to contribute to obesity during adulthood. So alterations of genes like leptin or insulin have been shown to contribute to obesity during adulthood. We also know that the environment is playing a role. So we know the diet, not doing a lot of exercise, microbiome, stressful situations can also contribute to obesity. And there is increasing evidence showing that chemicals are also important in this puzzle. And this idea of chemicals contributing to obesity challenges our previous understanding of obesity, because we used to think that it was all about diet and exercise. And it also challenges industry, because that is actually the view that they are trying to that is actually the factors that they are focusing in, and not thinking too much about how other factors like chemicals are contributing to this problem. We also know that the environment we are exposed to during utero is important in obesity later in life. So poor nutrition or smoking can lead to obesity in the offspring. And now we also know that the environment our ancestors were exposed to can also contribute to metabolic disruption and obesity in future generations. So one example of a human study studying the effect of environmental factors contributing to metabolic disruption is the Dutch famine cohort. So this cohort comes from a period of time where in the Netherlands there was a shortage in food. This happened at the end of World War II. And there was a very dramatic shortage of food accessibility. So in this right graph, you can see this column that is representing the desired amount of calories that every individual should take per day. So during the war, that is represented in this x-axis, there was a reduction in the amount of calories. It was period of time where war was around, and it made food less available. But at the end of 1944 and the beginning of 1945, in this region of the Netherlands, there was even a more dramatic reduction in the amount of calories that these people were taking. So something that was very extraordinary is that there are really good records about this cohort of people that suffer this famine. So part of the cohort were pregnant women. And these women were exposed to the famine during preconception period, so right before they got pregnant, and also during pregnancy. And something very beautiful is that scientists decided to actually reach out to the offspring. And so those individuals that were exposed to the famine during development and even before conception. And they found an interesting pattern that a lot of these individuals had different diseases. So they were either obese or had altered lipid profiles, metabolic syndrome, diabetes, glucose metabolism, or even they had fertility issues or behavioral alterations. So they actually further this analysis following to the next generation. So this will be the grandkids of the mothers that were exposed to the famine. And in general, they showed poor health. So how does this happen? This is called a multigenerational effect. So these pregnant females, pregnant women, I'm sorry, I use female because I'm used to work with mice, but when we talk about humans, I should say woman. So pregnant women were exposed to the famine, and then that was reflected multiple generations later. So if we think about our previous understanding on how inheritance work, we tend to think about genetics. So in this context, we will have an environmental perturbation that will cause one particular mutation in the genome, and then by the power of genetics, we will have one particular phenotype. However, what we found, or what was found in this cohort, is that the environmental perturbation that will be the famine will alters something that is still to be defined, and that will lead to a panel of many different phenotypes, including metabolic disruption, infertility, or behavioral alterations. And it is thought that epigenetics is playing a role in this context. So what is epigenetics? So there are many definitions of epigenetics, and you could probably hear, I think every researcher is going to use a slightly different definition. The one that is currently accepted is the study of changes in organisms caused by modifications of gene expression without altering DNA sequence. So what does this mean? So when we think about our genome, when we learn for the first time in the school what our genome is and what is genetics and what are genes, so we start learning about the DNA helix. So we think about this fiber in which our genes are contained. And then these genes can be regulated, and depending on the expression, we are going to have like particular physical features, or in many occasions, we can develop certain diseases. This DNA is wrapping family of proteins that are called histones, and they can continue to get compacted, the whole nucleosome that is called, they can continue to get compacted so that this fiber can fit in the nucleus of the cell. And the nucleus of the cell is very small, so the DNA needs to get very, very well compacted. So by making these different loops and by getting more and more compacted, there are certain regions of the genome that are originally very far away, like it could be represented something like in here, that they get closer because they are making these loops. And then everything gets more and more complex as the compaction increases. And then what is represented here is the nucleus, and each of the colors will represent one chromosome. So what is epigenetics here? So we can see epigenetic modifications at different levels, and one of the modifications that we can identify are DNA methylation, which will mean that there is a methyl group that is added in certain places in the genome, and that is going to alter how those regions of the genome are going to be expressed. Then there are histone modifications, and it is more or less the same idea. These proteins can be modified with chemical alterations, and that is going to affect the accessibility of certain proteins to the region where these histones are and how the genome is expressed. And there is also chromatin compaction. So this mess that is represented here is actually not that messy. So there is some control in how this chromatin is compacted in the nucleus, and there are some regions that are more accessible than others, and that can also contribute to how the genome is expressed. So going back to our original question, in this cohort of the Dutch famine, it is thought that there are epigenetic mechanisms involved in this multi-generational inheritance of detrimental phenotypes, but it is still unclear what is actually going on. So the work that I do in the lab is partially inspired in these kind of studies. So we want to understand how other factors, in this case, we want to understand endocrine disrupting chemicals, how they are. The first question was whether they contribute to multi-generational disease, and if so, what is the mechanism? So when I started this project, I'm sorry, I need to drink some water. So when I started this project during my postdoc, we wanted to understand whether endocrine disrupting chemicals could lead to transgenerational obesity. And we started working with this chemical, tributyltin, or TBT, which is an antifouling agent that can be found in paints and wood preservatives, and is also a thermal stabilizer. It has been found in house dust and human blood, and that it tells us that humans are exposed to it. And it was in the past used to paint the boats, and if the paint contained TBT, that would prevent barnacles, microorganisms, and other mollusks to grow or to attach to the boat. However, there is a strong regulation right now with the use of tributyltin, and the result is like is shown in this image, where you can see that the boat is pretty damaged by all these organisms that are attached to it. So something that was known when I started working in this project is that TBT induces lipid storage in cell culture. So when we expose cells to differentiating stem cells to differentiate into adipocytes that are fat cells, if we expose them to tributyltin, they accumulate more lipids that are represented here in red. They accumulate more lipids than in the control that will be represented here by the MSO. These two samples are not important in this discussion, but definitely tributyltin induces more lipid accumulation than the control group. And it was also known that TBT induces fat tissue development in mice. However, there was no information about whether or not TBT induced transgenerational obesity. So I started the project, and what I did was to expose pregnant females, what we call the F0, to tributyltin, and then we track multiple generations. So in this context, since we are exposing pregnant females, we cannot discard the possibility of having the first generation being exposed to the chemicals, so that will be the embryo. And also the second generation. So in the embryo, there are these cells that are called primordial germ cells that are going to become spermatocytes and oocytes. So they are going to lead to the F2, the second generation. So in this context, the F1 and the F2 could be potentially exposed to TBT. So we decided to look into the F3 and later generations, because the F3 will be the first generation that for sure we knew that was not exposed to tributyltin. So we used mice. We used this mouse strain called C57BL6J. The short name is Black 6. We use 15 nanomolar tributyltin in drinking water, and there are a couple of important points here. So we use drinking water because it is the route of exposure that humans are thought to be. Oral exposure, I think, is not necessarily drinking water, but oral exposure is the route of exposure that humans are thought to be exposed to tributyltin. And the concentration that we were using is 15 nanomolar TBT because it is lower than the NOAL. So if you remember my very first couple of slides, the NOAL is a no observed adverse effect level. So we were using a concentration that is lower than what industry will call safe. And yeah, again, this concentration is also relevant to human levels found in blood. So what we did was to expose the animals and then the pregnant females, and then we tracked multiple generations. And when we reached the fourth generation, that was for sure not exposed to tributyltin any time, what we did was to expose these animals to what we call the diet challenge. So generally we keep the animals on a standard diet. And what we did with these animals is to, for a period of time, change the diet to a high-fat diet and then return the animals to the standard diet. So in terms of humans, this will be something similar to a healthy diet that we are eating during the summer and fall season. And then eventually we have the holiday season, and for a few weeks we get a little distracted and we don't eat that healthy. And then we, after holidays, we want to go back to our healthy routine to get ready for the summer again. And so we will do something kind of similar to the animals. And what we found is that in the case of males, that is represented in this graph, the animals that were ancestrally exposed to TBT, that is represented here with this dotted line, when we change the diet from the standard diet to the high-fat diet, these animals accumulated significantly more fat than the control animals that are the DMSO animals that are represented here in the solid line. So it is a very big difference in terms of fat accumulation. And then when we change the diet back, both of them lost some weight. It would be equivalent to a diet, but the animals ancestrally exposed to TBT didn't recover as much. We also analyzed females, and in this case, we don't see data that is statistically significant, but there is a very nice trend, and further analysis are needed to actually determine whether females are showing a similar phenotype. Another challenge that we expose the animals to is fasting. So generally these animals are fed ad libitum. So they have free access to food. They can eat anytime they want. And what we did in this experiment was to actually remove the food of the animals for a few hours. And we measure body composition, fat content, in the animals. And then we calculated how much fat the animals lost during the fasting period. So here DMSO again is the control, and these are negative numbers. So the control group mobilized more fat than the TBT group. So this means that ancestral exposure to TBT is somewhat hampering fat mobilization. Again, we look, this is in males, and we looked also in females, and we don't see a statistically significant result. However, the number of animals that we had here was slightly lower than with males. So we need to do like a larger, another experiment with a larger N number. So hopefully we can determine better whether or not there is an effect here. Another important point that we identified is that TBT alters the metabolomic fingerprint in both males and females. So in the case of females, as I showed you before, we didn't see a strong effect in terms of fat content. But when we look at the metabolomic profile, we actually saw a very strong phenotype in both cases. So what is this? Metabolomic profile refers to all the different metabolites that we have in our blood, and that includes glucose, insulin, all these different molecules that are helping our body to function. So each color here represents a different treatment. Green is DMSO, light blue is low concentration of TBT, this is 5 nanomolar, and dark blue will be 50 nanomolar. And basically what you can see here is that all these three groups separated really well. They don't overlap. So that means that they are very different. And that is as much as we need to know right now. And then in the case of females, we actually see the same pattern. So DMSO, that is the control, separates really well from the two TBT treatments. So even though we don't see any effect in females in terms of fat content, in terms of the metabolomic profile, females are also very different. So females are also affected by ancestral exposure to tributyltin. So the next question will be, okay, this is great. We have exposure of the F0, that will be the great-great-grandparents, and 4 generations later, we have descendants that are fat, but how does that happen? So we decided to look into the molecular mechanisms, and we isolated fat tissue from these animals, and we did analysis of DNA methylation, that is one of these epigenetic modifications that I described at the beginning of the talk. And we also looked into transcriptome, which is the overall expression of the genome, how each of the genes is expressed in these animals. And we did this in the animals that were ancestrally exposed with TBT, and with DMSO, that would be the control. And the overall conclusion is that there was an alteration of nuclear genome organization. I'm not going to get into a lot of detail into that, but what does this nuclear genome organization mean? So I want to go back to this slide where I am showing how the DNA is very, very heavily compacted in the nucleus. So nuclear genome organization refers to how the genome is compacted in the nucleus. And as I said at the beginning, it seems that it's pretty messy and pretty random, but it is actually not. There is a little bit of order in how this genome is in the nucleus. So there are two compartments that can be identified here. So there is what is called the compartment A, that is made out of what is called euchromatin. And euchromatin represents active regions of the genome, which means that the genes that are located in those regions are more actively transcribed. Compartment B, represented here in red, represent heterochromatin. And this region of the genome has genes that are not being expressed, and it tends to be located in the periphery of the nucleus. So this is just like an ideal way of representing the two compartments of the nucleus, but everything is a lot more complex. Everything in biology is a lot more complex than whatever model we want to figure out. So for instance, this is the previous image that I just showed you in the previous slide, but if you look at different cells, you're going to see a slightly different pattern. So yeah, the heterochromatin, that is the red component, is in the periphery in all these examples, but you know, there is a little bit of heterochromatin in the center here, so the distribution is slightly different. Then there are other different cell types where there is a lot more heterochromatin than euchromatin in terms of the proportion, and there is also variation depending on the cell. And there are other cell types where actually the euchromatin is in the periphery and the heterochromatin is in the center of the nucleus. And one example is the rod cells in the eyes that are in charge of detecting light in darkness. So, you know, depending on the cell type, there are alterations, and even within the same cell type, you can see variation. So what we think is happening is that tributyltin is actually altering how this chromatin is localized in the nucleus. And we don't think the changes are very dramatic. We don't think it is changing from here to here. It might be something closer to this type of alterations that is biasing the cells into a particular direction that can actually lead to alterations on how the genome is expressed in a way that is going to contribute to metabolic disruption in the overall picture of the individual. So after that, we decided to also look into how this information is propagated from one generation to the next. So our hypothesis is that the alterations of this nuclear genome organization occurs very early in development, like right at the beginning when this F1 generation is being developed. So then how do we go from this first generation to the fourth? We think that, so we did some analysis, we analyzed different tissues in different generations in both sexes, and we also did DNA methylation and transcriptome. And the conclusion we raised is that there is a self-reconstructive propagation. And I don't want to go a lot into detail into what does this mean, but this is important because until this time, nobody had been able to actually study this type of propagation of alterations after exposure to environmental factors. So what is next? One of the projects that we are working on right now is in trying to understand what is happening at these very early stages when the man is being exposed to the pregnant woman. The pregnant female is being exposed to tributyltin. And actually one hypothesis that we have, and it is based on preliminary data, is that something happens before conception when the adults are exposed to tributyltin that is going to alter either the maternal biology, like their metabolism, or the oocytes that are going to be fertilized with the sperm that are going to lead to the first generation. That is actually the initial step that will contribute to transgenerational obesity. Another project that we are working on is trying to answer whether these mechanisms are shared with other environmental factors. So as I mentioned before, we are exposed to many different factors that have many different natures. So I have talked a lot about toxicants and pollutants that we are exposed to, but I mentioned before that there is like the diet, there is the social component, and other factors, and all of them interact with each other. So it is important that we look at these questions from a more holistic perspective, rather than focusing on only one chemical. Try to understand how other factors may be interacting with each other and whether they have similar mechanisms of action. So in that project, we are actually working with arsenic, that is a naturally occurring chemical in the environment. So there is pretty much everybody in the world is exposed to arsenic at more or less levels, and it has been associated with metabolic disruption. We also want to look into high-fat diets. So the diet is not a chemical, but it's also a factor that contributes to obesity. So we want to understand if factors that have different nature lead to similar results in similar ways. And actually one project that we are working right now on is trying to analyze the interaction between these two. Instead of analyzing them separately, we are analyzing them together. I don't have any data yet because we just started, but I'm very excited about this project. So the third project that I would like to do right now, we are not working on it, but it's something that I would like to pursue in the future, is whether different individuals from the same species respond similarly to the same environmental exposure. So something that we are learning with this COVID crisis is that not everybody that is infected with COVID suffers the disease in the same way. So depending on your underlying conditions, depending on other health problems that you might have, you may experience COVID in a very different way. So something that I would like to do is to actually test the effect of the same environmental factors in 3 different mouse strains. So they are the same species, but they are genetically different. So we cannot, if we use the same mouse strain, we will be using like an identical genomic background. And in this case, we are trying to introduce some genetic variation, some genomic variation. And I chose these 3 different mouse strains because Black 6, that is the mouse strain that I have been working with for a long time, is susceptible to metabolic disruption. AJ is resistant to metabolic disruption, and 129 is neutral. So it will be interesting to see whether the same chemical induces similar phenotypes and what are the mechanisms. And finally, the last project that we are developing is to actually study whether different species respond similarly to the same environmental factors. So this work is inspired by many different things, but there was this paper back in 2011 where the researchers analyzed many different group of animals that represented 10 different species. And they found that in the last 30 years, they gained significantly more weight in parallel to the weight gain that humans have observed. So in this graph, basically what is represented is the percentage weight gain, and this is the obesity status. So for instance, in this case, that are male and female chimpanzee, they are the most, the species that have evolved into a more overweight species in the study that is represented here. And you can argue that, there are some animals here represented like dogs and cats, that these animals live with humans. So yeah, it's likely that, your dog will eat your leftover pizza or something like that. But there are some animals here, like the male and female mice or rats, that are lab controls. So these animals live in they are represented here. So these animals live in very, very well controlled environments. So we regulate the diet that they take, the water that they take, the temperature in the room, how many animals are put in the same cage. We regulate absolutely everything. However, we feed them with a diet that is made with different grains that are grown in the crops around us. So these grains are treated with pesticides and other environmental factors that may very well affect the mice that are going to eat this food, and that can actually be a factor that is contributing to these animals to increase their body weight. So working with different species is very challenging. It's something that we cannot easily do in the lab if we want to work in vivo. However, we can use an in vitro model. So for that, we want to work with mesenchymal stem cells, or MSCs. These cells can differentiate in different cell types, depending on the stimuli they receive. And one of the cell types is actually fat cells. So what we want to do is to differentiate these cells, and we can use our control chemical, that will be DMSO, and we can use TBT. And in the case of TBT, we will see that there is an increase in the accumulation of lipids that are represented in this picture with this bright red color. So we can use MSCs from different species, and those are available in different companies that so we can buy them, grow them in the lab, and then expose them to different chemicals, and then see if these cells accumulate more lipids depending on the chemical, the factor they are exposed to. So some chemicals that we want to try are arsenic, nicotine, and PFAS that are all very prevalent in the environment and that have been associated with metabolic disruption. And I think it's a good way to start actually looking into how different species respond to environmental factors. So I hope with this talk, I gave you a little bit of overview of what we do in the lab, what is kind of our big picture, holistic perspective of the problem of environmental factors and metabolic disruption. And actually, I have talked a lot about metabolic disruption, but you know, there are, as I mentioned before, there are many other health concerns that are associated with environmental factors that are also very important to keep in mind. And that actually, it will be interesting to see in any of the experiments that we are developing in the lab, if they are actually altering other functions like the immune system or behavior, or you know, any fertility, any other aspect that is important in this context. And with that, I would like to thank my lab. So we are a small lab, but we are growing. We have Carlos, that is the genome master. He's the one that knows everything about nuclear genome organization and the one that came up with the idea of TBT having this effect. Stephanie is working in the arsenic project, and she just started a new experiment, and we don't have any data, so I couldn't show anything, but we are really excited about it. Catherine Palmer is rotating in my lab, and she's working with MSCs. I am around doing stuff. And we are part of the microbiology and environmental toxicology department at UCSC. We are part of the Institute for the Biology of Stem Cells, and I am a member of the Earth Center at UCSF. And we have some funding. Without money, we cannot do anything. So, and it mostly comes from UCSC, my startup, and a core grant that I got to work in the MSc project. And with that, I will be happy to take any questions. I saw that there is a
Thank you so much for your talk.
Thank you.
Let's open it up to 10 minutes of Q&A. If you don't want to speak using your microphone, just put it into the chat window. It looks like we do have a question from CP. And I will, Raquel, you can go ahead and read it. I'll read it out for everybody else. So considering bioaccumulation in animal product concentrates, many environmental toxins, maybe we should eat less of them. She read a large study about Inuit populations in Alaska that had a big problem because of toxins in their diet. which included birth defects, cancer, diabetes. Davis also studied this in plants and food, and a large number of negative effects were found in children. So she's saying that our environment is very polluted, and maybe eating low on the food chain is the way to go. Can you comment on these issues with our food supply?
Yeah, so definitely there is a lot of information, a lot of research being done with that regard, and everything points in the direction that animals like meat, so that these chemicals, most of these chemicals can bioaccumulate in animals. So if we most of these chemicals are also lipophilic, so this means that they can I don't know what is the word, but they accumulate in the fat. So any animal that has fat is likely to have these chemicals accumulated in their tissues. So if we then eat this meat, then we are going to indirectly be exposed to these pollutants. So there is definitely research done in that direction, and we should definitely think about the effect of our diet or the decisions that we make in terms of our diet and what is the impact in our health. Yeah, that's absolutely correct. There is also a lot of information about fish. So not only in meat versus fish amounts, but in the water. So we know that the oceans are polluted. There is all this trash in the ocean, and as it degrades, all these chemicals are released in the water, and then the animals that are in the water are exposed to it. And all these big fish like tuna, I mean, we know a lot of mercury, but it's not only mercury. There are many other chemicals that are accumulated in these kind of animals. And then if we eat it, we are exposed to these chemicals. And so it's definitely something that we should think about if we are concerned about these issues.
Raquel, I've got a question for you.
Mm-hmm.
So, so you showed that these transgenerational effects, persist from generation to generation, even out to F4. If you went further, would you expect that there would be reversal back to homeostasis, or is there a possibility that this is the new homeostasis?
So that needs to be tested. So I get that question a lot, and actually Carlos in my lab, he would love to do that experiment, like to have like a colony of animals that we keep forever and then see what happens. It is a very difficult experiment to do, but there is some, I mean, there are alternatives. There are other model systems that can be used, like a Drosophila or C. elegans, that their life cycle is shorter. So in those cases, it could be more manageable. So reaching a four generations with mice takes like 1 year. So if we want to keep them going for many generations, it's going to take a long time. It is very expensive, totally worth it, but we need an agency that is going to help us funding that research. So there are alternatives. We can use other model systems. There is research done in C. elegans where they exposed the worms to BPA, bisphenol A, that is another chemical that is very prevalent in the environment. And they found an effect in the first generations, and I think they tracked up to 14 generations. And after 7 generations, they started to lose the phenotype. So it is a little complicated to actually translate that into mammals. Worms are very different. They are a great model for certain things, but there is a big difference between the biology of the worms and the biology of mammals. There is also a big difference in epigenetics, methylation levels, and so drawing conclusions out of that experiment need to be made with caution. So it could be that we go far in many generations, and then we see that the animals adapt. But it will be necessary to do that experiment. And I think there is one researcher, Michael Skinner, in Washington State University, that I think is working in something like that, but the data is not published yet.
Yeah, so that's a great answer to the question. Thank you. Hopefully you keep those mice, and hopefully you learn something before I graduate, you know, in a few years, you know.
Well, I hope you graduate before I because otherwise that would mean that you're gonna be here for a long time.
For a while, yeah.
So yeah, no, that will take like a significant amount of, I mean, I don't want to talk about time as an element. It is something very interesting to do, but we need to be realistic about time, funding. I mean, if we don't have funding, these experiments are pretty expensive. So, and they also require a lot of attention. We need to keep track of all the mice. We need to, you know, be very careful of how we do all the matings and how we generate this generation. So it requires a lot of maintenance and attention. And so, yeah, I don't know, hopefully in the future, when I get more established at the university, in the department, and I get a lot of funding, maybe I can actually afford doing something like that.
It seems like somebody is talking about how people can financially support this work in the chat.
Yeah, so, okay, yeah, this is Branwyn. Yeah, I know her. I will definitely contact you. Thank you for saying that. It will be great to have some more funding. Yeah.
Awesome. So you're funded through the NIH, correct?
So right now, the funding that I have comes from the university. It is my startup funding, but I am applying for grants at the NIH.
That's awesome.
Yeah, so there is the National Institute of Environmental Health Science, where my you know, my research it fits really well. So I'm trying to get some funding from there, and I'm just reading that yeah, Branwyn, what Branwyn meant is that if anybody wants to fund my research, to contact her. So I think I misread what she meant. So yeah, that would be awesome. If anybody has that capacity, it would be really great.
Yeah, excellent. Do we have other questions from the audience? I had another question if nobody wants to speak up. Having to do with the cell-to-cell variation in epigenetic state.
Mm-hmm.
That was a pretty interesting slide to me, because at least thinking about it, the way I've been thinking about epigenetic states is that there's like these discrete programs, you know, like certain cells will be kind of locked into a particular program, like of expressing certain genes. And between the cells, I hadn't thought about how there could be variation in the epigenetic states. And is that reflective of there being like programs within programs, or is this kind of like a natural variation that can't really be fit into those kinds of boxes, you know?
I think it is a natural variation. So when we think about things like epigenetics, or any type of mechanism or any type of program, we need to simplify the ideas. So we need to, in order to be able to tackle those proxies, we need to be able to simplify them. We need to focus on one particular element. That is why, you know, when we are in the lab, we work with only one chemical or with only one factor. So we are able to narrow down what is happening and how is this happening. In terms of epigenetics, or the idea that we have about nuclear genome organization is that each cell is slightly different. And that is something that we have learned with the experience that we have in this kind of project. When we are, for instance, working with stem cells in vitro in a plate, we have multiple clones of the same cell that is very similar. These cells are very similar one another. So it is more likely to find very similar patterns in that context. However, if we are working in vivo, each cell is slightly different. Just the location within the organ is going to be different. It's not the same being in the liver in one lobe versus in a different lobe, or being in the fat tissue, in the surface of the fat tissue, or if it is inside the fat tissue that is going to have less oxygen, is going to have less accessibility to it. So there is going to be variation within the same cell type, within the same tissue. So we need to keep all that in mind when we are talking about these kind of things. It's not one fits all.
Mm-hmm.
So, and actually, something that I have seen with research that other people are doing, or collaborators that we have, is that sometimes if you are looking for the exact same behavior from animal to animal, or from, even within the same tissue, that doesn't happen. You don't see the same exact response. So it is important to keep all this variation in mind. And I think it's biological variation. It's not a program within another program, a model within another model that can explain.
Mm-hmm.
Yeah.
It doesn't just go forever.
Yeah. But it's true that, there is a lot of research done in vitro. And the power of that is that you have clones. So all the cells are going to respond very similarly. So you're going to have a very strong effect. And that's okay. But then when you jump into analyzing something similar in the whole organism, if you don't see that response, that doesn't mean that you invalidate your hypothesis. That means that you need to keep in mind variation.
Yeah.
And I know that Carlos could talk more about it. So, and he's probably trying to keep himself from saying anything.
I mean, it's something to think about. At least when I work with human cells, I'm like, oh, this is in vivo, but really it's not. It's a tissue that's in a dish. I'm controlling all these variables, and it's, as much as I like to think it's representative of an organism, it only gets me so far.
Actually, I mean, there is a lot of discussion into what is in vivo, in vitro. So ex vivo, yeah. Carlos is not here. So for the rest of the audience, Carlos is my husband and he works with me. So he's just in the living room, really close to me. So it is important that we use the right way to refer to these studies. So when you are taking cells from an animal or from a human, that is ex vivo. So you take the cells and then you culture them. And that can relate a little better with what can happen. But for instance, if you take cells only from one human, that is going to be different compared to another one. So you always need to test a number of different individuals to actually account for that variation.
Mm-hmm.
And I see that there are more questions here.
Oh, yeah.
So Gio is saying, in your last slide, you had temperature as a chemical physical factor that can affect humans. Do you think global warming could potentially affect our gene pool? Yes, and that is our interesting question. Actually, there is a lot of work done in other models like Drosophila, that I know that you're familiar, Gio, with it, and C. elegans, working with temperature. And, in humans, it's a little bit more difficult, but as the Earth heats, we are going to learn more and more about what is the effect of this global warming in our health. So I don't know exactly how, but I think definitely there is an effect on that. Yeah. For instance, I mean, it can be also not only a direct effect, but also indirect. So there is global warming. So there are certain bacteria, certain pathogens that are more comfortable growing in this warmer environment. And that is going to actually affect our health in a different way. So there are different direct and indirect ways global warming can affect us. And then CP is saying the EPA doesn't require the kind of testing you did before they approve new chemicals, pesticides, compounds, etc. Why not? I guess different chemicals interact with a multitude of chemicals in the environment too. Yes, exactly. So the EPA does require some control, but the criteria is not necessarily the same as the criteria we are using in basic research when we are trying to narrow down mechanisms. So the EPA uses measures like the low AL or the no AL to actually decide whether a chemical is concerning or not. And actually, there are efforts from the EPA to collaborate with basic scientists to actually test this hypothesis. However, I have a little bit of experience with that, and things are not very clear or very those kind of interactions are generally difficult because if our research is showing something that is unexpected, then it's very difficult to actually argue. I mean, we need to use our data, and that is very powerful, but we need to really use a strong evidence to prove to the EPA that one particular chemical should be removed from the market.
And I think just to add to this conversation, a lot of new chemicals, the burden of proof of toxicity is not on the EPA sometimes. Like they can release a chemical, they can synthesize a new molecule, be it like a dye or whatnot, and the EPA doesn't have to test it until it's proven to be toxic already. Isn't that true?
Exactly. Yeah. So that is where there is miscommunication between agencies, regulatory agencies, and scientists. And I think something that is very important is that there are multidisciplinary collaborations. So, for instance, chemists synthesize chemicals. So it is very important that when a new chemical is made, that they communicate with biologists, so we can test different models and we can test, in different species or in whatever research we are doing, whether these chemicals are having a detrimental effect. However, one difficulty there is that if there is a new chemical, there is little information about it. So our capacity to get funding to actually develop a thorough experiment using complex animal models or research models is very reduced. So, it's like a balance. We need to first define that there is a problem, and then we will address the problem. And one example was, for instance, with BPA. So bisphenol A, it was very obvious many years ago that it has a detrimental effect. So it was, there was a lot of pressure to remove it from the market. So they removed it, but they used substitutions like BPF and BPS. So that would be bisphenol F and bisphenol S, that chemically are very similar, and probably you know more about this than I do. But so in my mind, how is it possible that you don't test a chemical that is very similar to a chemical that has been proven that it harms our health? How is it possible that it is put in the market? So we are going to be exposed to it, and it's going to take years to identify that this chemical is actually having a detrimental effect in our health. So yeah, more research is needed, and more collaborations are needed.
Yeah. Well, thank you for the work that you do. I hope that the government takes notice and, implements this kind of strategy, on a larger scale, maybe grouping classes of molecules together if they share some sort of similarities together before they release them to the market to just increase the throughput of how they test,
Yeah, there is a little work on that, but most of the work is done actually in vitro or ex vivo. And as we have discussed, it has some limitations. So yeah, we need to be mindful of all that.
Yeah. Do we have any other questions from the audience? Okay. Well, Raquel, thank you so much.
Thank you all. I appreciate your time and your attention and all the questions, all the really great questions.
Yeah, that was great discussion.
Yeah.
So we will be back next month. We're going to have Science on Tap a week earlier than usual because of Thanksgiving celebrations. So expect to see the flyer quite soon about the date and the topic for the talk. So Raquel, thank you again so much. I'll see you back on campus.
See you around.
Yeah, see you around. And we'll see you all soon again. Alrighty.
Thank you all.
Oh, to John, yeah, we are recording talk and I'm going to upload it to YouTube pretty soon. So you can catch the first part. Don't worry. Alrighty.
Thank you.
Bye, everybody. Have a good night.